Hello. Good evening, everyone. No, that was not me playing the piano. Welcome to the Live Lab here at McMaster University. My name is Joe Kim, and I am the Academic Director of the McCall MacBain Postdoctoral Fellows Teaching and Leadership Program. This is a program dedicated to creating professional development opportunities for postdoc scholars from around Canada. And one of our fundamental goals is to provide early career researchers with tools to improve their knowledge translation skills and to be able to convey their research impact, which is exactly what we'll be doing this evening. I would like to acknowledge that the land in which we are currently gathered sits on the traditional territories of the Mississauga and Haudenosaunee nations, and within the lands protected by the Dish with One Spoon Wampum Agreement. It's important to note that teachings from the First Peoples of Canada have had an enormous impact on all areas of our academic pursuits. I'd also like to acknowledge and thank the people who have helped make this evening and our program possible. Marcy and John McCall MacBain are visionaries who are committed to building the next generation of teachers and leaders. Thank you for your continued support and friendship. Tonight, you are seated in one of the most unique research centers and performance spaces in Canada, and it's called the Live Lab. The large interactive virtual environment lab is a unique 106-seat performance hall designed to investigate the experience of music, dance, multimedia presentations, and human interaction, and it will host our amazing research talks tonight. So I just want to show you a couple of the features. So right now, you are listening to me speak in a room as if it's a classroom setting, but we're using virtual acoustics. So let me show you a couple things. So if I just clap my hand, you can hear the echo. But at the touch of an iPad, I'm going to change the acoustics of this room so it's a much larger room. And we can use this space to do all sorts of interesting research. So for example, if we wanted to do research about distraction and attention, if we're testing out hearing aids in a busy restaurant setting, I could add in this noise and we could see how that might distract people. There's some subtle, this is one of my favorite ones. Let me add in. Might as well bring a helicopter in too. We're not going to be looking at that tonight. So we're just going to keep this as a regular classroom space. So the space where you're sitting right now includes active acoustic control, sound recording equipment, and measurement of behavioral responses like movement through motion capture, brain responses through EEG, muscle tension through EMG, heart rate, breathing rate, sweating responses through galvanic skin response. We're not measuring any of those tonight, but this entire theater can be reconfigured to measure directly from the audience all of these different types of electrophysiological measures. So I'm going to show you a brief video that will tell you a bit more about this unique space before we get started.
This is the first time I've ever experienced anything like this in my entire career of 10 plus years. The complexity of the kinds of sounds they can achieve is enormous.
Hello, everybody. Welcome to Live Lab. It's great to see you.
Well, it's actually about 8 years in the planning from when we initially had this idea to going through the building of the facility to now when we're actually ready to open. We have amazing musicians. We're going to demo the virtual acoustics and all around have a fun time.
The facility is absolutely unique in the world, first for its sound quality, which is really quite excellent, as well as this active acoustic system that lets us create any acoustic environment that we want. We have a number of systems that let us record responses from both musicians and audience members. Their heart rate, their respiration, brain waves, so that we can figure out what's going on inside of their head while a performance is going on.
When they adjust the acoustics of the room with their computers, you can hear the effects that it has on the music itself, and it's an extremely powerful experience.
We have a long history here of very, very innovative, groundbreaking research in sometimes unexpected areas. And this is another classic example of that.
The thing that blows me away is just the connection between things like music and performance and science and brainwaves. Every time we talk to someone about the Live Lab, we hear, well, I could use it for that or this. It seems to be never-ending possibilities. So I think this is just the beginning. Lots of possibilities in all sorts of different fields. So it's really exciting.
Thank you very much, Joe. Hello, everybody, and welcome to the Live Lab. My name is Katie George, and I'm the Program Manager for the McCall MacBain Postdoctoral Fellows Teaching and Leadership Program. One of the longest titles on campus, I think, maybe. Pardon me. I want to welcome everyone to what promises to be a really amazing night. We hope that you have enjoyed some delicious food, and now we're going to give you some appetizers for your mind. This evening, we will have the privilege of hearing from 7 elite researchers from 5 institutions around Canada. They are the finalists from the McCall MacBain research competition entitled the 5MRT, or 5-Minute Research Talks. This also included 30 researchers competing for the top spots. So tonight, you have the privilege of hearing the top 7. They were tasked to translate their complex research and communicate it in just 5 minutes to an interdisciplinary audience. And I'm not sure if anybody is familiar with any of their research, but that is a very large task. These are individuals that work in very niche areas, very specific, very elite, and they're able to take that knowledge and convey it to anybody. And that is really a deep skill. I want to thank all of the fellows tonight for all of their hard work in preparing their presentations. Their innovation, impactful research, and dedication to their work will inspire you as you listen to each 5-minute talk. At the end of the evening, we'll have a brief question and answer period with all of the researchers. So we ask that you keep any sort of lingering thoughts in your mind till the very, very end, to the last speaker. You will also have an opportunity to vote. So this particular area will be decided on popular vote. So your votes matter. So as you're listening to the talks, take note of what impressed you, what was impactful, and any of those lingering questions. At the end, we're going to award a top prize of $500 to the winning researcher who gains your votes. Now, a little caveat. You can't just log in and vote for your friends. You have to ensure that you actually assess each talk. And this is why we're waiting to the end to assess everybody's votes, okay? So without further ado then, I'm going to get the evening started. and introduce you to our first speaker, Dr. Ben Sclodnick from McMaster University. Dr. Sclodnick studies the contributions of memory and learning to visual attention in humans. He has a passion for science and believes that the future of humanity depends on scientific thinking. To that end, he regularly takes on teaching and leadership roles that involve interacting with people and supporting their academic development. Ben is also the founder of the popular Psychology Live public science talks, hosted locally at Fairweather Brewery. Outside the lab, you can often find him correcting people's grammar, riding his motorcycle, or playing the guitar. So, without further ado, Dr. Sclodnick, and I'm going to take my paper so he can't see my grammatical mistakes.
All right. Backwards. There it is. Okay. I'm going to talk to you about measuring attentional control. So, whether you're reading, driving, sitting in a lecture theater, maybe witnessing a crime, or even just scrolling on your phone, attention determines what we see, what we remember, and what we act on. If we want to understand human behavior at large, and of course I think we should, we need to understand the factors that control attention. So let's see if we can identify some of those factors together. We'll start with the demo. Everyone look at the screen. Okay. First thing I want you to notice is that your attention is probably drawn to the spiky explosion thing in the bottom corner of the figure. Okay? We call this saliency-driven attention. Things in the visual field that are physically distinct gain automatic priority over attention and draw our attention to it. Okay? That's why we put brake lights on the back of vehicles and why police cars are so annoying. Okay? The second thing I want you to notice is a little more subtle. Take a look for one of those blue boomerangs. And once you've found one, notice how the other blue boomerangs in the display seem to stand out a little bit more than they did before. Okay? We call this experience-driven attention. And it shows us that paying attention to something in one moment biases attention towards those things in future moments. And already we can put together a little model of attention here. We have physical saliency, experience-driven attention, and of course, the thing that you get to control, your goal-driven attention, your conscious control. It was you that looked for the blue boomerangs, right? Okay. What I'm interested in are the effect of experience on attention, these experience-driven effects. As a field, we're realizing that these effects are ubiquitous across our behaviors, and they're also challenging to measure. And they're challenging to measure because they're often confounded with our goals. Okay? And so, take a look again for one of those blue boomerangs. Okay, now you have your conscious goals driving you towards the blue boomerang, but you also have the influence of experience guiding you towards the blue boomerang from having just looked for one. And so already you can see that you have two attentional factors driving your attention to the blue boomerang, and it's difficult to disentangle those effects in an experiment. Okay, so this is me. My whole program of research is based around finding clever methods to disambiguate the role of experience-driven attention from goal-driven attention. And I'm going to give you an example of how we might do that. So in my tasks, I typically use a visual search task where the target is defined by physical saliency. It's the odd color, the odd one out. And this target could be blue, or the target could be orange. And your task as a participant is to find the target, the odd color, and then just indicate which corner of the diamond is missing with the key press. Then, in the context of an experiment, we can get people prepared and ready to expect blue things. We say, get ready for blue, right? And so we have blue targets and orange targets for our search task, but we fixed goal-driven attention. And so we should be able to measure an effective goal-driven attention here. And sure enough, if we look at response times to blue targets and orange targets, you can see that people are faster to respond to blue targets than they are to orange targets, okay? And so that's all well and dandy. But of course, in this experiment, people are preparing for blue on every trial of the experiment. We know when people repeatedly prioritize the same thing, well, that's what leads to those pesky experience-driven effects on attention, right? And so to control for the effective experience, what we need is another group of participants that prepare for blue on one trial, prepare for orange on the next trial, prepare for blue on the next trial, and orange, and so on and so forth, to control for the effect of repeatedly preparing for the same thing over and over again. When we do that, we can see that our effect of preparing for blue gets smaller when we intermix preparation, right? On the left is larger because that represents the influence of both goal-driven attention and experience-driven attention. Where on the right, we've controlled for experience-driven attention. And that difference is a measure of experience-driven attention on performance. And so that's a sneak peek of how we can use some clever methods to differentiate the influences of experience and goal-driven attention on attention. And this is critical because whether you're a radiologist who's repeatedly scanning x-rays for particular blotches, if we want to understand that behavior, we need to understand the factors that control attention. And that means we need to understand a way to measure the effect of experience on control. And the same is true for the tourist in London who's about to walk out onto the road looking the wrong way. And the same is true for any of us who find ourselves repeatedly battling social media for control over our attention. If we want to make ground on understanding these behaviors and making a difference, we need to understand how to measure the factors that control attention, starting with the role of experience. Thank you.
Sorry,
Clicker.
I interrupted the bow. Thank you, Dr. Sclodnick. That's awesome. Thank you. Now I'll just click it to our next presenter. Next, we have Dr. Thomas Kazmirchuk from Carleton University. Dr. Kazmirchuk studies AI-assisted drug design. He became a scientist to help people. His current work focuses on the development of therapeutics and diagnostics for bleeding disorders. Not only does it help him channel his passions, his research will also have a very real-world impact on improving patient care as well as quality of life throughout the world. Here's Dr. Kazmirchuk.
Thank you so much. Passion, like blood, unites us. Some of us apply our passions to creating beautiful music, like we just heard. Others construct wonderful structures, like the Live Lab. More still, apply their passions to curing diseases. I apply my passions to improving patient care and quality of life for people with bleeding disorders. In doing so, I'm designing the future of medicine. We get injured, we then bleed, and then we stop. This process takes about 15 minutes and is due to coagulation and clot formation. However, not everybody can do this process effectively. In fact, every 1 in 4 deaths across the world are due to some sort of bleeding or coagulation disorder. What's more, 1 in every 10,000 people are affected with some sort of bleeding, clotting, or coagulation disorder. It is these people I am seeking to help. How? At Carleton University in Ottawa, we've developed an artificial intelligence that can design small molecules that can act as therapeutics and diagnostics to protein-related diseases. How it works is we take our protein of interest and we put it into our AI. The AI then intelligently designs small molecules that are predicted to interact with our target protein. At the same time, the artificial intelligence will discard any of these molecules, design molecules, that display off-target or cross-reactivity. In this way, using this AI, I designed 23 molecules to a platelet protein that, when mutated, causes a platelet disorder that is difficult to treat and even harder to diagnose. Now, I'm going to tell you 2 results that highlight this work. First, I'm going to tell you about a synthetic clot. So in blood and in this experimental system, a green mutated platelet protein will interact with a blue blood protein. I'm looking for a molecule that disrupts that interaction and thus will prevent or disintegrate diseased clots. So what will happen is this magenta molecule will interact with the green platelet protein and block its interaction with the blue blood protein. And this is exactly what we see. In this highlighted image, you see two pictures. On the top is a synthetic clot, and this is just in saline. And then with our magenta molecule, on the bottom, you see that this aggregate, this clot, is completely disintegrated. And this is quantified on the far right, and this just shows us how potent and how effective, at least in the lab, our magenta molecule is. Now, I told you that clotting takes about 15 minutes. The other major question we had is, does this molecule that works, does it carry out its function in this time frame? In other words, what I did is I incubated the green platelet protein with the blue blood protein like this for 15 minutes. I then took that reaction volume, split it into two equal volumes, supplemented one with saline and one with our magenta molecule. The goal is to see whether our magenta molecule strips green off of blue. This, again, is exactly what we see. I'm showing you a highlighted image in a red box where you see a black blob. The black blob is indicative of the green and blue proteins interacting in saline. But when supplemented with our magenta molecule, you see nothing in the lane right beside it. This shows that the green and blue proteins are no longer interacting with each other. And this is a 100% ablation. This molecule is extremely potent. To that point, our molecule interacts with the green mutant platelet protein 15 times stronger than COVID binds to our cells. That's how strong of an interaction this molecule has, and its affinity is remarkable. Now, in the next few years, we're going to test this molecule in patients. I'm very excited to tell everybody here and online that we just got some results yesterday about the effectiveness of our molecule on patient-derived platelets from Italy. Our molecule was able to restore normal function of the mutant platelets in a diseased system. This is remarkable. We may have translated our molecule out of the lab and into real life. So, building on this, we're going to then test our molecule in mice, and furthermore, we're going to develop more diagnostics with our industry partners in Halifax and Hamilton, and with the goal of curing and treating and diagnosing diseases. For this point, the molecule I presented here, it is the first treatment and potential diagnostic for this platelet disorder ever. Now, I hope you can see that we're truly designing the future of medicine. In other words, it's in our blood. Thank you.
Thank you so much, Dr. Kazmirchuk. What an amazing presentation, and thank you for your dedication to such an important and impactful cause. Next, we have Dr. Idowu Olawoye from Western University. Dr. Olawoye is a microbiologist and bioinformatician, took me a while to get that one, with particular interest in understanding bacterial pathogens, their evolution, as well as antimicrobial resistant patterns. To do this, he uses computational tools to analyze genomic and epidemiological data to provide insights to public health agencies. An interesting fact about Idowu is that he is ambidextrous, which is a trait that he shares with only 1% of the population. So without further ado, Dr. Olawoye.
Hi, everyone. Just a few years ago, I completed my PhD studying the bacteria that causes tuberculosis, which in fact is a slow-growing bacteria. It's hard to grow in the lab and experiment on because it's highly infectious and it can be drug resistant. But now it's time to move on. And when microbiologists like myself switch to studying new bacteria, they would either study a new bacteria in a different bacteria that belong to the same family, or switch to something completely different, maybe something easy where they can grow in the lab and it's fast growing and they can plan experiments from day to day, which would lead to scientific discoveries and obviously scientific publications. But I've done the complete opposite of this. I've decided to study something that is difficult to grow in the lab, is equally transmissible, and it can be drug resistant. Allow me to introduce to you MGen, which is also known as Mycoplasma genitalium. This is a sexually transmitted infection that can infect both male and female, and it disproportionately affects the marginalized groups. And this bacteria, if left untreated, can actually lead to things like infertility or even cervical cancer. And you might be wondering, why have I decided to take this route? Like, why did I decide to study something that's hard to grow? And that's because I'm trying to catch it before you or someone else you know catches it. And because it's an important bacteria, we need to be able to test it in the lab. And it's quite daunting. It's a difficult thing to do. One of the challenges is because it's extremely slow growing, which means most bacteria usually take a day to grow in the lab. A slow grower like Mycobacterium tuberculosis will take about 6 to 8 weeks. But for these, will probably take about 2 or 3 months if you're lucky. Also, it is a fastidious bacteria, which means it's very picky. Imagine you have a toddler who's picky with food. That can be quite frustrating. And it's the same with this bacteria. You need to replicate or mimic the human host conditions for it to grow, which can be quite challenging. And also, it's resistant to most antibiotics that are used in treating this disease, and that's because it lacks a cell wall that most bacteria have. And because it doesn't have this, these drugs will not target what's not present. And like I said, it's a very important bacteria. It's a very important disease. It's not quite known. It's understudied, and that's because it's really hard to study. It's hard to grow. But with the current trends, there are more infections spreading which are difficult to treat. And one of the detection methods is called, is done using PCR machines, which is an equipment like this. And this is a benchtop equipment, which, oops, don't know what's going on. It's not responding, oops. Yep, I think that just froze. Okay. So it's a benchtop equipment like this, and it usually will require additional equipment like a computer or gel imaging device, and for additional analysis. And it uses variable temperatures to amplify the DNA of the bacteria to know if it's present in your clinical sample. And it can usually take about 2 to 3 hours to get your data, which can be quite long if you're not really skilled to do this kind of analysis. And it's pretty expensive also, which means labs in low-income countries or under-resourced area would have challenges in procuring this. So what my research is doing is to take a new technology called LAMP assay, which doesn't require the large equipment and all the secondary devices for analysis. What you just need is a heating block like this, so you can literally procure these on eBay. And what it does is it allows for colorimetric detection, which means you can visualize the result with your eyes without needing computers or anything. And it uses a constant temperature to amplify the DNA of your bacteria. And your results can be ready under an hour, and it's cheap and easy to use, which means we can deploy this in under-resourced areas. And this is what your result will usually look like when you have a PCR analysis. You could have graphs like that might be quite difficult to interpret, or a gel image of that nature. But with what we've been able to do in the lab, this is the results we achieved. You can see there's a color change from red to yellow. Yellow is where we have a positive sample to MGen, and the red one is a negative control, which means you can literally see the change if the sample, if it's positive or not. And the overarching goal of my research is to improve STI testing. And because we know time is of the essence, we need to be able to test drug resistance rapidly, which will then prevent the progression of this disease to its chronic stage and improve health equity to everyone. And I would like to appreciate our team and our funders, and thank you for listening.
Thank you so much, Dr. Olawoye. And thank you for all of the work that you do. We're very grateful that we have people like you to do such important jobs like this. Our fourth speaker of the evening is Dr. Myanca Rodrigues from McMaster University. Dr. Rodrigues works as this is another fun one, biostatistician. People often expect her to focus only on the numbers, but her real passion is what is behind the numbers, the stories. In studying mental health and addiction, she realized that opioid use disorder isn't just about overdose deaths. Many people live with its long-term deficits and effects. Her work uses statistical methods to uncover the trajectory of illness and to find ways to improve their quality of life. Without further ado, Dr. Myanca Rodrigues. One moment, please. Okay, a little bit of a switcheroo. I apologize. So you will see Myanca, I promise. She's waiting in the wings and she's ready to go. But our PowerPoint wants to give you Sina first. So I'd like to introduce you now to Dr. Sina Baleka from McMaster University also. Her research explores ancient DNA from extinct elephant species to reconstruct their distribution and admixture patterns. She then connects these insights to past climate events. She originally studied anthropology, and the ancient DNA lab had some elephant samples that no one really wanted to work on. And everybody wanted the human samples, of course. So she kind of fell into this type of work. I guess it goes to show you that one person's passion is another person's unwanted elephant samples. So here she is, Dr. Sina Baleka.
So, does anyone here know what a Colombian mammoth is? And I'm saving you right, because it's really complicated. Let me explain. The steppe mammoth evolved around 1.7 million years ago in Eastern Asia, and it migrated to Europe and lived in Eurasia during most of the early and middle Pleistocene. It also migrated to North America, and somewhere along the line, it evolved into the Colombian mammoth, a species that is adapted to a warm climate and has been distributed from the north of the United States all the way down south to Costa Rica, with the earliest samples being around 1.2 million years old. That sounds relatively straightforward, but there's more. In Siberia, the steppe mammoth evolved into the woolly mammoth, the only mammoth species that is adapted to an Arctic climate. And with the advance of the next ice age, the woolly mammoth also migrated to Europe as well as North America. So it's still not too complicated, but nature is messy, and we can see this when we look at the DNA of these extinct species. Paleogenetics, or the field of ancient DNA, works with DNA from samples that can be hundreds or hundred thousands of years old. It comes with a suite of challenges. Most times the DNA is completely degraded and there's nothing left to analyze. If you find DNA, then it's heavily damaged, fragmented into very short pieces, and contaminated with everything from modern human DNA to ancient microbial one. So most of the DNA sequence that we have today are less than 100,000 years old. And this is true for all of the Colombian mammoth sequences that exist, and also for most of the woolly mammoth ones. The oldest paleogenomes that we have today come from two steppe mammoth sequences from Siberia that have been dated to around 1 million years. Dinosaurs, 65 million years that direction, so we do not have any DNA from them. Now, even with that DNA from the Colombian mammoth, we can see that the species in the late Pleistocene is actually a hybrid between a woolly mammoth and a lineage coming from the steppe mammoth from Siberia. But you do not know where and when this hybridization took place between a species that is adapted to a warm climate and one that is adapted to the exact opposite. Where did they meet? To answer this question, we would need more DNA from the North American continent that is older than most of the DNA that we currently have. Change of topic, Arctic ground squirrels. Cute, right? Arctic ground squirrels lived in northern Canada for hundreds of thousands of years. They dig into the permafrost where they build their burrows called mittens. We we recently analyzed one of those mittens from the Yukon that has been security dated to 700,000 years ago. And to be more precise, we analyzed Arctic ground squirrels' paleofeces, and that just means old poop. Now, the amazing thing when working with feces, and you do not hear that sentence a lot, is that you don't just get the DNA from the defecator, but also from everything that they ate, giving you a lot of information about the ecosystem at the time. Now, Arctic ground squirrels are not strict herbivores. They also eat insects, small mammals, and might feed on carrion of larger mammals. So in our Arctic ground squirrel paleofeces, we found mammoth DNA, 700,000 years old from North America. Analyzing this DNA and comparing it to everything that's available, we could see that at this point in time, we do not see any hybridization. So to further narrow down when this hybridization took place, we will need additional data. But why is this interesting? Genetic data from extinct species is more than a scientific curiosity. It reminds us of what we lost and gives us insights into how and why this happened. Understanding the evolutionary histories of extinct species helps us understand how these species reacted to certain stresses in the past. This can help us act instead of react to extinction threats for our living species. Thank you.
Thank you, Dr. Baleka. I had no idea that you could learn so much from poop. My kids will be thrilled. Now I'm just going to ensure that it is the proper speaker. Yes, the woman that needs no introduction now, Dr. Myanca Rodrigues.
So I will be talking to you about how we have to move beyond overdose in the opioid crisis. Every day, 21 Canadians die from opioid overdoses. But what about the 350,000 who survive and live with opioid use disorder? Meet one of them. Alex struggles with depression, complicating his treatment. As he comes off opioids, the issues that led to his use remain unaddressed. What's worse, he has heart disease. People with opioid use disorder are at a higher risk of heart disease and heart attacks compared to the general population, particularly as they're recovering from opioid use. And Alex isn't alone. Opioid dependence affects the entire body, with many people often experiencing 2 or more chronic diseases, known as multimorbidity. Yet, the overdose problem is almost synonymous with opioid crisis, and few studies examine the long-term health impacts. Which brings me to the bigger issue. Despite Canada being at the center of a worsening opioid crisis, we do not have a lot of research looking at the long-term health impacts of this disorder. Without this data, we lack the knowledge to design care that works. And this isn't just a medical problem, it's a health system failure. Multimorbidity leads to greater complications, higher costs, and places the entire health system under strain. So how do we fix this? That's where my research comes in. To address this gap, I used Ontario as a case study, a province with both rural and urban treatment sites, diverse socioeconomic conditions, and a representative sample of the crisis. I analyzed data from over 3,400 people receiving treatment across 57 addiction centers between 2011 and 2021, linking their clinical records to provincial health data and following each person's health data for 8 years. First, I identified 16 chronic diseases that are commonly included in multimorbidity research. Next, I assessed their prevalence over an 8-year follow-up with the bubble size corresponding to how common each disease was. Then, among those who had at least 2 chronic conditions, I applied hierarchical cluster analysis to identify disease clusters based on how often conditions co-occurred. So what did I find? 1,114 people in the sample had multimorbidity. That means 1 in 3 people with opioid dependence has at least 2 other chronic diseases. And among this subset, 6 distinct disease clusters emerged, the most pertinent of which I will discuss today is the largest cluster comprising cardiovascular, cardiometabolic, and respiratory diseases. Now, this affected over 660 people and requires very careful consideration. For example, if a patient with opioid dependence additionally has COPD and congestive heart failure, well, this poses some severe risks. The ACE inhibitors for heart failure can interact with opioids, causing dangerously low blood pressure and breathing issues. Opioids further suppress breathing, which complicates COPD management. Now imagine this patient navigating a health system where specialists don't coordinate care. The risks are enormous. That's why we need a multidisciplinary approach where primary care, cardiology, and addiction specialists work together to provide safer, more effective treatment. Canada, one of the countries hardest hit by the opioid crisis, still lacks critical data on its long-term health impacts. Without it, people like Alex continue to fall through the cracks. My work lays the foundation for a new model of care, one that doesn't just treat opioid use disorder, but recognizes and addresses the full scope of health challenges that people face. Because the goal isn't just to respond to crises, it's to move beyond overdose. A huge thank you to my study participants, supervisors, the McCall MacBain Program, and thank you for listening to me.
Thank you so much, Myanca, for all of the really important work that you're doing. Our final speaker for the evening is Dr. Ahmed Shakeri from the University of Toronto. Pardon me. His research interest integrates advanced biomaterials, microfabrication, and surface engineering to develop biomimetic, yes, tissue models and diagnostic devices. Try saying that 5 times fast. Dr. Shakeri notes that he was always fascinated by the idea of building organs outside of the body, almost like something out of a science fiction movie. The ability to observe cells in action within miniature bioengineered organs is incredible. It can help us study disease more closely, uncover innovative treatments, and develop solutions with a lasting impact for human health. Here is Dr. Shakeri.
Hi everyone, my name is Amit and I'm really excited to share my research on the heart's hidden shield against cancer. Cancer has been the 2nd leading cause of death worldwide, but nowadays, thanks to advances in medicine, many types of cancers can be treated effectively. But the treatment gets challenging when cancer spreads in the body, a process called metastasis. When this happens, the cancer cells dislodge from the initial tumor, circulate within the bloodstream, and invade other organs. They can spread to the stomach, for example, liver, and other organs. But here's something strange. Cancer almost never spreads to the heart. Think about it. Has anyone ever heard of heart cancer? I really hope not. So this is a very rare phenomenon. But why is the heart so resistant to cancer? This is the question I'm investigating in the radistics lab at the University of Toronto by modeling cancer metastasis outside of the body on a chip. Now, to model cancer metastasis, the first step is to create a porous tube so that the cancer cells can get into and spread. Next, we place this tube in a platform with 5 chambers, and the chambers are only connected together through the tube. Then we grow a mini version of human organs inside these chambers, for example, heart, liver, and breast cancer. Now, to stimulate blood flow, we place the entire platform on a rocker with gentle tilting motion, causing the media to go back and forth through the device. To create the porous tube, we use 3D printing with a coaxial needle, which is basically two needles, one into the other one. Now, through the outer needle, we perfuse a biocompatible polymer called POMAC, but in the shape of granules or microparticles to get the porosity. Through the inner needle, we perfuse a temporary gel just to keep the tube open during the printing, and it will be washed away after. As the materials get out of the needle, we shine it with UV flashlight to basically solidify it. I coined these tubes permeatubes because they can mimic the permeability of blood vessels. Here you can see how we use a 3D printer to precisely print these tubes. And here is how the final version of the device looks like. Looking at the permeatubes at higher magnification, these SEM images show the topography and cross-sectional of the tubes. You'll notice there are two types of porosities. There are bigger pores with a size of around 40 micrometers, and inside them there are smaller pores with a size of 2 to 4 micrometers. So we started out by studying liver metastasis. We first grow the liver tissue on a chamber on the right, and then once the cells are established, we introduce a highly aggressive breast cancer cells into the middle well. Here you can see the permeatube in blue and cancer cells in green, how they want to enter the porosity of the tube to spread. Next, we track the cancer cell migration over time. So we use a fluorescent microscope where only the cancer cells and the tube were visible in green. Day 0, right after introducing the cancer cells, you can see all the cells are in the middle chamber as expected. On day 6, you can see a few of them have already migrated to other chambers. And by day 10, they're fully migrated across the platform. Next, we repeated the same experiment, but this time we included the heart tissue. Here you can see a video of the heart that we created out of stem cells. You can see how nicely it's beating. This little heart is only 2 millimeter by 2 millimeter in size. Apparently, it's too small for a Valentine gift, as my wife said. But anyways, we tracked the cancer metastasis in this platform as well. And on day 0, as expected, there was no metastasis. Day 6, we still didn't see any metastasis. And even by day 10, there were barely any cells metastasized in other chambers. So why is the heart so resilient against cancer? One reason could be attributed to the proteins that are secreted by the heart, which can prevent cancer cell migration. We, in fact, are going to dig deeper into the tissues in our platform to analyze such proteins. Who knows, maybe one day we can come up with a drug that is inspired by the heart environment to combat cancer. After all, the heart might not just beat for us, but it might also hold the key to fighting cancer. Thank you for your attention.
Thank you so much, Dr. Shakeri. What a fascinating subject. Now we're going to switch gears a little bit for our final speaker. They're not with us today in the room. Our final speaker actually joins us from the beautiful province of Quebec. Dr. Romain Villot arrived in Quebec City in 2015 to do his PhD. He studied a new transcription factor involved in cellular senescence triggered by oncogenes. His current work involves new molecular mechanisms that regulate cellular senescence, especially in retinopathy. Research in this area can lead to new avenues to treat age-related disease and help people to live longer and healthier. Outside of the lab, Dr. Villot often is involved in some kind of sport, whether that be weight training, running, and skateboarding. So here he is via Zoom, Dr. Romain Villot.
Hello everyone, hello. So thank you so much for the invitation, and I'm really sorry to be not present in live, but it's a pleasure to talk about my postdoctoral project. And I'm glad to discuss about the concept of cellular aging, especially cellular senescence, and how studying cellular aging could help treat vision loss. So as we age, many disorders and diseases, such as eye disorder, become more common, and it is the case of the retinopathy. And retinopathy is a disease of the eyes that happen when blood vessels of the retina, which is a really important part of the eyes, are defective. causing a deficiency in nutrient and a lack of oxygen or hypoxia. So the retina, to try to survive in this really bad condition, trigger a phenomenon that we call pathological angiogenesis. And pathological angiogenesis refers to the growth of little and really fragile vessels that invade the vitreous, invade the eyes, leak bleed in the eyes, and at the end impact a lot the vision. That is interesting is that in this disease, retinal cells enter in cellular senescence. And cellular senescence is a specific state of arrest of the proliferation triggered by many different stressors, such as DNA damage that we accumulate over time. So we accumulate senescent cells over time, and these cells are known to contribute to tissue dysfunction and many aging-associated diseases. So senescent cells have many characteristics, but especially these cells are known to secrete and release a lot of different molecules with many different functions. And some of these molecules are known to promote, to enhance the pathological angiogenesis and the retinopathy. Indeed, it was shown by different groups that the elimination of senescent cells or the inhibition of their secretory activity could decrease pathological angiogenesis and slow down the progression of the retinopathy, clearly showing that these cells play a crucial role in this disease. So that's why it is really important to understand and to decipher the mechanism, the molecular mechanism that drive cellular senescence, notably during retinopathy. So for that, I will just take 1 second to remind you what is a gene. So a gene is a segment of DNA that contains the instruction to make a specific protein. Thus, Paul Lemire, that's why a former postdoc in the lab of Frédéric Antoine Mallette, used a retinopathic mice to perform gene expression analysis in order to identify new senescence effector and eventually develop targeted therapy. At the end of the analysis, we observed that one enzyme, named CH25H for cholesterol 25 hydroxylase, increased a lot during retinopathy. And we observe more recently that this enzyme also increased during cellular senescence because we have protocol to induce cellular senescence in vitro. For example, we can use the irradiation that cause DNA damage and induce cellular senescence. And you can see on this image of Western blot, which is a really good technique that we use to evaluate the abundance of the protein, that the enzyme CH25H increased a lot during cellular senescence. And this result shows that the elevation of CH25H is not only a feature of the retinopathy, but could be a universal hallmark of cellular senescence. But what is CH25H and what do CH25H? So it is, as I said, an enzyme that change the cholesterol in another molecule that we name the 25-hydroxycholesterol. And we observe that when we expose many different cell lines, notably retinal human cell line with a really low amount of 25-hydroxycholesterol, we observe that these cells enter in cellular senescence. As you can see with these cells with the blue color, which represents the beta-galactosidase activity, which is one of the best ways to identify senescent cells. So, in conclusion, we observe that the enzyme CH25H is increased during retinopathy and cellular senescence and promote cellular senescence, notably by the production of the 25-hydroxycholesterol. And because cellular senescence is known to induce pathological angiogenesis and aggravate the retinopathy, we think that developing pharmacological molecules that target this enzyme could be a promising avenue to treat retinopathy, but also maybe other aging-related disease. So I would like to thank you so much for your attention. I would like to thank all my lab, especially Paul Lemire and Frédéric Antoine Mallette, my PI, to allow me to work on this wonderful project. And thank you so much again.
Thank you, Dr. Villot, and thank you for joining us online. What an amazing subject, and you've definitely ended our evening on a very intriguing note. At this point tonight, I'm going to call forward all of the speakers from this evening. If you could come up and watch your step. Join me on stage. As the speakers are joining me on stage, it's time to look at your little notepads and look at those lingering questions or thoughts that the speakers might have triggered for you. There we are. Got everybody? All right, we're all here. Yay, gang. All right, so any questions for our speakers can be answered now. Pardon me. All you need to do is raise your hand, and a volunteer will assist you with a microphone. I would also ask that you take this opportunity and time to vote for your favorite talks. Go online. Let me get the QR code going. You all have access to the QR code, or you should. And it's also here. So you can find it on your, oh, there we are. It is also on your schedule for the evening. So you can find it at the bottom, or you can use the code in front of you for those online. Everybody has the opportunity to vote now. Make sure that you go in and assess all of the individuals that have spoken this evening for your vote to count. All right. Pardon me. So we will open up the questions for the audience.
Thank you all for such insightful talks and the work that you're doing. My question is for Dr. Shakeri. I was kind of confused about the image showing both the liver and the heart cells. How come the cancer didn't spread to the right side? I understand why it didn't spread to the heart.
Sorry, that's a really good question. So we are actually investigating this. This was also interesting to us. I think the main reason is probably the cytokines, the proteins, and the EVs that are secreted from the heart tissue. Because when we put the platform on this rocker that I showed, the media gets mixed really easily, really quickly over time. So whatever is secreted by the heart tissue can go into other wells. It's actually preventing cancer migration to other wells as well, which was very interesting to us. It's not just only the heart tissue, but other tissues as well. But at this point, we are not sure yet.
Thank you.
Thank you all for the great talks. Very interesting. My question is for Amit Shakeri. So the heart that you've shown, is it just a replica of the heart with the exact same, everything the same, it's just smaller, or no, it lacks some of the components, some of the parts? And then the follow-up is that how far we are from just growing a heart in the lab from stem cells and just transplanting to a human?
Yeah, so for this part, it for sure doesn't have the complexity of the natural heart. But what we did here was we differentiated iPSCs. There are induced pluripotent stem cells that are actually got from like human skin, I believe. So it's cardiomyocyte cells that are differentiated from human stem cells. And then to create a tissue, we mix the cells with cardiac fibroblasts to make it as close as biomimetic as to the actual heart, but like it doesn't have all the preferred orientation of the cardiomyocytes in the heart. But like these cardiomyocytes can spontaneously beat as well. That's why you could see the beating. It doesn't mean that it can actually be transplanted at this point into the a patient. So regarding your second question, I think we are pretty far yet, but there have been like very huge advancements in that regard. I think the main issue is creating a scaffold that can accommodate for such cells, so that when we can transplant them into the patient's body, they can, I guess, withstand the structure for the time.
So this is a question for Dr. Rodrigues. How do you think some of the challenges in terms of creating this like multi-dimensional treatment could actually like go down in Ontario itself, especially like with political challenges with funding and training and whatnot?
Yeah, that's a really great question. I really hope Doug Ford has some answers. But I would say that in terms of what could work, and maybe I'll answer the question that way. I think, so I know the way that psychosis programs work, early intervention and psychosis programs. Now, those often are targeted to people below the age of like 30, 35. And so as soon as you're diagnosed with it and you get into like CAMH or any kind of psychiatric treatment facility, well, they appoint you a patient navigator to follow you through the system. And so they connect you with like social work, a nutrition, because often, you know, there's social factors, there's lifestyle behaviors that are also the target for that, in addition to psychology and psychiatry. And so I feel like if you know, a program like that exists with allied health professionals in addition to medical professionals, and there's some kind of integration working, it's facilitated by this patient navigator. And so how much more easily can we not do this when we have all medical professionals, right? Like we have OHIP billings at the end of the day that connect all these providers. So I know that there is a code now, diagnostic code in the billing system in some way, perhaps not the billing system, but there's a code that captures multimorbidity. So perhaps providers can look at that and flag that when they're treating their patient at the point of care. And in terms of how it could work is we know that, you know, they're going to these addiction sites. That's where we've recruited them from. Well, at that point, maybe you can get a patient navigator or some kind of referral to medical services, and then making sure that they get screened and tested and going through the system and ensuring that their medications kind of take into account the other conditions that they have. And I guess that would be my ideal world answer. And in terms of the challenges, funding, as always, and having some kind of way to remunerate healthcare providers for participation in this system.
Amazing, thank you.
Okay, thank you all for the great presentation. My question is to Dr. Idowu Olawoye. In comparing the PCR and the newly developed diagnostic setup for the MGen, is it only qualitative, like color-wise, or does it have any quantitative value for the new one? Is one concern. The other one is, did you really check the false positive, false negative aspect of that newly developed setup? Thank you.
That's a very great question. So in terms of the question number one, so it's not just qualitative. Obviously, you see the color change, you know, but we are currently looking at how sensitive it is. Like, what is the least amount of DNA sample or amount of cells that, you know, the LAMP assay can detect. So currently, we are optimizing the assay. We're able to like see a detection rate of about I think about 200 cells, which is quite small, but we're still trying to like, you know, optimize our assay to make sure the detection level is at the minimal threshold, which is comparable to PCR, because you know PCR is the gold standard. So we're trying to at least match what PCR can detect, and that would be, you know, the best case scenario. Then also in terms of the false positive and false negative, we are currently trying to get isolates from, you know, the microflora of someone who has an STI, because obviously, you know, when you do this kind of test, there's a chances where
your assay might pick up other things and detect this as a false positive. So we're currently trying to optimize our assay to make sure we have, isolates from the genital microbiome, and then we can use that to see if our assay picks these other organisms and sees it as a false positive. So we're currently just trying to optimize the assay to make sure it's robust enough to be deployed out there, but we're not there yet. So it's still a work in progress because this is a very slow-growing bacteria. It's hard to study. It's a bit of a challenge, but we are making some good, progress in that respect. Thank you.
Thank you. I have a question for Dr. Kazmirchuk. I was wondering, what does the timeline for your molecule look like? And then also, can you use the AI that you use to develop cures or molecules for different diseases?
So, great questions. For the timeline, I'll just tell you that our initial designs for this molecule occurred in January 2023. And so now we just showed, literally yesterday, it works on patient platelets. So usually at this stage, these molecules, whether they're what I work on or they're antibodies, they usually take around 6.5 years to get to this point. We've done it in 2. So the timeline's been accelerated significantly. We're now looking to go into mice, and that'll be a couple years. So I'm going to say, assuming everything works, knock on wood, and everything's promising and we publish, maybe we can start to talk to the FDA in certainly the next 5 years, if everything works. That's a big if. And then the second question was about what is the applicability of this technology to other diseases. Was that correct? Okay. So proteins cause many diseases like cancer, like diabetes, like bleeding disorders. And so what we can do is our technology, we can design these therapeutics and diagnostics to any protein-related disease. If you think about COVID, we developed a diagnostic for COVID using our technology, and we actually have manuscripts for a therapeutic for COVID. My work is for bleeding disorders. We have works for cancer proteins. We have work for other pathogen mitigation technologies with the government. And so we have collaborations with the University of Toronto and some in Halifax. So if it's a protein-related disease, we can do something with it.
Thank you for all the great presentations. I have a quick question for Dr. Ben. So regarding the attention, so is there any difference between children and adults? For example, my daughter are curious about everything. So I'm thinking, is there any theory to explain this? Thank you.
Yeah, that's a great question. I don't study children. And for the most part, a lot of the research that's done on attention is done with undergraduate students at university. I don't know off the top of my head theories about children, but yeah, I'd have to talk to somebody who's an expert in development. Why kids are interested in everything? Because they're still learning, they don't know what everything is. Things are still novel, right? So for us, things we get really used to things in our environment with lots of exposure, and things become less interesting. So I think with less experience with things, when there's something that's new in the environment that you haven't encountered before, those things tend to capture our attention, and we can measure that in adults. That's one of my best guesses off the top of my head as to one of the things that might influence children more than adults, or at least influence children to be interested and fascinated and attracted to things that we think are mundane.
There's a question online for Dr. Kazmirchuk. I'm curious to know how many candidate molecules you started with, and how long did it take you to narrow it to the ultimately selected move to the clinic?
Perfect. Thank you for that online question. We designed initially 23 of these molecules, and I sort of answered it earlier. I'll just maybe reinforce it a bit. These initial designs came out around January, February 2023. And so, 2 years later, we're now seeing an effect on patient platelets. That's extremely quick. And then mouse model, this is the point where drugs die, when we start to translate out of the lab into the clinic. So, you know, we hope that we can get these studies done rather quickly, quickly meaning maybe about 2 to 3 years, depending on funding, depending on availability of the mouse model. But we're actively having these discussions right now, and we're actively trying to get the funds. If there are any venture capitalists out there who want to fund these studies, please let me know. So all that to say, we had 23 molecules initially. One showed this high potency effect that I presented, and we are pushing the timelines as fast as possible by working as hard as we can.
Just a quick follow-up. Is AI being used to help identify candidates?
Yes, exactly. So that the essence of our technology will actually design and select molecules that have high affinity to a target protein of interest. In this case, it's a mutated platelet protein. And so it designs the molecules and it selects for affinity and specificity. At the same time, though, we assess that the AI assesses that same molecule against off-target proteins, so like a cancer protein, like a cell protein. And if any of those molecules both interact with our mutated platelet protein and another protein not involved in anything, any process, the AI automatically discards that from any further analysis. We also the AI designs these molecules over thousands of cycles, so anything that is essentially makes it through is predicted to have extremely high affinity and specificity to the target, and then we go validate it in the lab. And of that, we found one that works very well.
Could also help write the paper too.
Thank you all the speakers for the great presentation. Actually, my question is for Dr. Thomas, and then the question just been answered regarding the AI application in the medicine design. So then I have one follow-up on that, more about the open discussion. So in your opinion, right, so are we going to see the revolution change in terms of medicine design by, using AI? If so, what would be the barrier?
Okay, so that's a fantastic question. Essentially, where are we going with AI in science and medicine? Is it going to take over everything? What is my opinion on that? I think that AI is a tool that will help us do all the menial tasks and all the tasks that we can't complete. For example, there are millions of scientific manuscripts out there. Not everybody can even be an expert in their own field anymore because there are more papers published than we can keep up with. A computer, an AI, can keep up with it. And so the breadth of knowledge that humanity is producing on a daily basis can be interpreted, analyzed, and contextualized by artificial intelligence at a rate faster than any human can. Everybody at this university, McMaster University, has a specific knowledge base. If you take everybody at McMaster and combine their knowledge bases, it will have no sort of, you cannot compare it to the breadth of knowledge of an AI. So I think that AI is going to be pivotal to discovery in science, designing molecules like what I presented here, and just to allowing us as scientists to ask even deeper questions. So with the computer, there was this idea that everybody's going to lose their job, librarians, all that. That didn't happen. Jobs got easier. Although some jobs had to go, we were able to ask even deeper questions. With the invention of the computer and its large-scale applicability to society, we were able to sequence the human genome. And that allowed us to ask even deeper questions. I believe, and my opinion is that the same thing is going to happen with AI, and it's already happening. It will allow us to ask deeper and more meaningful questions. And I think we should all embrace our AI overlords.
Okay, we'll have 2 more questions.
I wanted to say thank you again for the amazing talks. My question is for Dr. Baleka. I'm just curious, what's the process of actually collecting samples and finding things like pellets? Just especially with things like deforestation, I can't imagine it's easy to find relevant samples. So what's that like?
Yeah, thank you for the question. So we're generally not the persons collecting these samples, so we are in collaboration with museums or paleontologists. Especially in the Yukon, they do a lot of mining there, so they have people that mine permafrost, they actually thaw it, and these people that do this, they have been taught by paleontologists to look out for certain things. So they work together with them, and then they get called and be like, okay, look, we found this. Are you interested in that? That's like how we find these kinds of like for the ground squirrels, you find the whole nest. So it's not just like a fecal pellet. It's the whole nest where you find everything in. And then normally when we work with anything like bones or teeth, like that's what you find in every museum, where we just go to the museums and ask them, do you have this? Can we just get a little bit? So yeah, that's normally the process that we follow.
Thank you all. It was such an amazing night. Congratulations to everyone. I have two questions for two different presenters, if that is okay. Okay, the first one is for Dr. Thomas. And a really interesting topic, and I was just wondering how, maybe I misunderstood, but you mentioned your approach has been tested yesterday, having results in humans. Okay, and you also mentioned you are going back to mice to test it, right?
Correct.
So can you please expand a little bit on the ethical challenges, or not challenges, but implications regarding the phase 1, 2, 3, and 4 for the RCTs?
Absolutely. I'll do my best. I'm still new to clinical trial stuff. I will do my best. So the ethics behind creating something that's never been created before, putting it on human platelets, see that works, and then translating it into mice for an eventual, hopeful, therapeutic. What are the ethics behind that? So really, to me, it comes down to is there any negative side effects of that molecule to the patient, to the mice, or whatever? Part of clinical trials and safety is safety testing and dosage. That's why we go into the mice first, is we look for the effect of the molecule in mice, and then we also assess its dosage and its efficacy and its safety. And so if the effective dose kills the mice, well, then we are not going to move forward on that, right? And so the ethics of it, of using mice to get this far, I would say it's what is commonly said about this in rebuttal to that comment is these mice, they are bred for this purpose, but they are taken extremely well taken care of, and they're bred for that purpose. And millions of scientists across the world use mice for this purpose. Now, we actually won't be killing the mice because this is a bleeding disorder that affects the first 15 minutes post-injury, a clot. So all we need to do is actively monitor the molecule in the blood, which there are techniques to do that while the animal is sedated, so it'll feel nothing. And we can also do a technique called tail bleeding time, in which, again, we sedate the mice, and then we just nick their tail, put the tail in saline, and we see the effect of our molecule. This is a common approach used in bleeding studies. So we actually don't end up killing the mice. We use them for, acute studies in this case. And so part of their job, though, is to see, before we get into humans, whether the effective dose is lethal. Hopefully not, but that is their purpose. And so ethically, we have to clear all of the ethical, they aren't hurdles, all the ethical guidelines before we even can attempt to get into the mice. And this is discussions that are happening right now. Every lab on the earth takes care of their mice, takes care of their scientific animals so that they're in no pain and that we can use them ethically. There are professions that are dedicated to this, and this is my answer. I hope it was sufficient.
Thank you very much. Yeah. And my second question is for Dr. Myanca. A fascinating talk. And I think I totally agree with you with the use of community health workers and to help navigate patients and get them increased access to healthcare and things. And I just would love to hear what do you think about the community-based approach, getting involved the participants or getting involved the patient or the client into the protocol writing and just basically inviting them to be co-investigators.
So I think if I've understood correctly, you're talking about patient engagement in research. Yeah, so I'm really glad you asked that. So I know, like the lab that that I work with, they definitely do a lot more patient engagement in research. So they go into the clinics and they kind of try to identify patient partners that they feel would be good co-investigators or that are even interested in it. And I know now we're trying to design a fentanyl-focused program. I know fentanyl seems to be buzzword of 2025, and we're trying to see from patients and their perspectives. Well, firstly, why? What attracts you to doing drugs in the first place, right? Like, why are you using illicit drugs? And if maybe we hear it from their perspectives, we can design intervention programs that target exactly why they're doing it. So we're approaching it from that kind of intervention perspectives. In terms of multimorbidity and getting them involved in that sphere, it hasn't happened yet. So the study that I conducted, that was the first study in Canada that looked at multimorbidity in people with opioid dependence. A lot of our research does come from south of the border, but I do hope that in the future we can engage them more and therefore design intervention programs that don't just target opioid addiction, but also the host of chronic diseases that seem to go along with it.
Thanks so much. So I have one final question I just want to ask Dr. Sclodnick. There's a couple of interesting trending topics on social media about attention. One is that there's a surge of adults getting diagnosed for ADHD. And second, there's an idea that generations of children now that are exposed to lots of multimedia might be better at multitasking. So my question broadly is the widespread impact of digital distractions, do you think that's making us better or worse at attentional control?
So you asked two questions, sort of, talking about ADHD. I mean, one issue about ADHD, of course, is that we're getting better at diagnosing it. And the awareness or the ability, even the thought to diagnose it in adults is sort of something that's relatively new. So like, it's hard to know whether the rates of ADHD are going up or, of course, if we're just better at diagnosing it. As for whether experience, like, the role of social media is also interesting. So if you think about kids being exposed to social media and technology, it's possible that they get just better at using it. So like when my phone bombards me all day and it captures my attention constantly, it takes me off track. I like have a love-hate relationship with my phone. But for somebody who had that experience when they were a kid and a teenager, they might just like learn to cope with that and to expect it and to even just tune into that information and capture it much more quickly than I can just by having that during developmental phases. This is not something I'm an expert in, but I think it's something we need to learn more about because, yeah, social media is definitely changing the way we think.
All right, thank you so much to our speakers and thank you to our audience for such thoughtful questions. Katie George has been busy tallying and she is here to announce.
All right, so Joe, I'm going to make you my Vanna White if that's okay. I will take this. You can demonstrate. All right, so the votes are tallied, but before I announce a name of our winner, let's give a round of applause for everybody standing before you today. These individuals are conducting some very amazing work that will one day change our world. Thank you for all that you do, whether it be understanding human attention or the mysteries of the heart, unearthing the secrets of ancient poop, I mean DNA, or understanding the needs of vulnerable populations in healthcare. Postdoctoral researchers drive our understanding of the world. As a thank you, each finalist this evening will receive an award created by local artist Paul Rodrigues from Dundas, Ontario. These glass sculptures were created for tonight with the theme knowledge. Each takes the form of a glass circle, encasing the iconic McMaster colors. I find it fitting that the awards this evening were crafted in glass. Glass is the combination of millions of grains of sand that are fused and transformed by intense heat into something new, something useful. Much like the hours of dedicated research performed by this evening's speakers that culminates into the creation of impactful knowledge and innovation. Thank you to our early career researchers for creating the intense heat that is required to transform our understanding of the world. Now, without further delay, I feel like I need a drum roll. Come on, guys. Here we go. Oh. All right. That's the right one. Pauline is alerting me here. The winner is Dr. Ahmed Shakeri. Round of applause, everybody. Thank you. Thank you to everyone who made this evening possible, to the McCall MacBain Foundation for believing in the researchers and leaders of tomorrow, to the McMaster Office of Alumni Engagement, and to our amazing researchers, of course, and to all of you for joining us this evening. We hope that you have a safe drive home. Thank you so much.